Hello, welcome. I'm Anna Galelli, and we're going to continue working on part 5 of our muscle physiology, and we're going to hone in on the resting membrane potential and what that means. All right, let's look at what we're talking about here. I'm going to draw a muscle cell. All right, and it's a really dumb looking muscle cell. So this is your muscle cell, and this is the sarcolemma. Okay, now the sarcolemma, remember, is your plasma membrane. So when we're talking about the resting membrane potential, we're talking about the plasma membrane. We're talking about the sarcolemma. And by potential, we're talking about the energy or the voltage on either side. So if I kind of blow this up, those of y'all who've checked car batteries or whatever, you may have used something called a voltmeter. All right. And this voltmeter has those little cords that come out, and you got a little thing that goes right here. And then we're going to take another one and we're going to put it right here. What you're doing to measure voltage is you're measuring the change from one side of the membrane to the other. And it's the same idea as what you're doing with a battery when you're using a voltmeter to look at one side versus the other, okay? So you're looking at the difference in charge on the inside versus the outside of the cell, okay? And this is what we're doing when we measure the voltage. If you average out all the charged ions, everything's going to kind of come out to the same thing. But when you're looking at a cell membrane, what you're going to find is that you've got concentrations. And these concentrations are going to be different on either side of the membrane. So if you remember on the previous video, I talked about how you have a 3:2 ratio of sodium to potassium on either side of the membrane. And really, whether it's sodium or potassium doesn't matter. What matters is the fact that they are both positively charged ions. And if I look at the outside versus the inside, I see that I'm always going to have more positive ions on the outside than the inside. And you know what? We do have negative ions in here. We got these little chloride ions that are negative, but we're not even worrying about them. We're just looking at this ratio of 3:2, which means the outside is going to be more positive and the inside is going to be less positive, which basically means it's positive on the outside and negative on the inside. Not because there are negative ions making the inside negative, okay? It's just because there are fewer positive ions, okay? So the inside is slightly more negative than the outside. In order to create what is called our action potential, you need to change the polarity, okay? And when you change the polarity, you either depolarize it or you repolarize it, okay? And when I say polarity, I am referring to the membrane potential, okay? Or resting membrane potential. So at rest, you have this 3:2 ratio. If I make the inside more positive, I have depolarized it. If I make it even less positive, then I am repolarizing it. Now, these things are co-occurring as you are changing the membrane potential, okay? Because basically you can kind of think of let me change that. You can think of repolarization as chasing depolarization whenever it occurs. Okay? All right. Now the action potential is kind of a special sort of thing. And we're going to talk more about this as we go along. We're going to talk more about it with neurophysiology. But first of all, an action potential is an all or nothing generation of voltage change. Okay? Now, this is going to occur at those voltage gates along the sarcolemma. Now, here's a little preview from neurophysiology. The action potential is started by what is called a graded potential, which is also sometimes called a local potential. Okay? This is started at the NMJ when the acetylcholine gates open and allow sodium to come in. So that's a depolarization that does not self-propagate. It is solely dependent on the opening of the acetylcholine gates and allowing sodium to come in. So it just kind of stays there. And the goal is that this graded potential will reach the voltage gates, and that will then generate an action potential. So that's a little bit of a preview. Let's go ahead and look at a little picture with some plus and minuses on it. All right, looking here at this diagram that I've taken from a book, I've got my sarcolemma and I've got my sarcolemma. And what we're showing here is that there are 3 sodiums to every 2 potassium on the inside. I still have potassium on the outside and I still have sodium on the inside as well. It's just the overall ratio. And what's happening is that even though these are both positive ions, I've got more pluses on the outside than I have pluses on the inside. So therefore, the outside is positive and the inside is less positive or negative. Okay, so that's resting membrane potential. Now here in this picture, what we're showing is that we have opened up a gate, and that has allowed sodium ions to flow inside the cell. What happens then is I've now got more positively charged ions on the inside of the cell, so that takes this little section of membrane and it depolarizes it by making it more positive on the inside. Now you will see it's still positive here and still negative there. Okay, so this is depolarization. If you are dealing with the motor end plate and chemical gates, this is called a graded potential. If you are dealing with the rest of the sarcolemma and this is opening up voltage gates, it's called an action potential. And basically this is going to chase its way down all along the membrane. And we can see that in the next couple of pictures where you can see that it's basically chasing. So you allow sodium in, that changes the polarity here, and it just keeps on going and going and going. Now, as it's going, you're actually going to begin repolarizing the back by opening up by allowing more potassium to run out, to rush out, okay? And that causes repolarization. So that's what this is showing you, is like you've got initial depolarization, sodium comes in, all right? If you're at voltage gates, it propagates and you create an action potential. As you are doing that, the part that just depolarized begins to repolarize because potassium is going to start rushing out. And this is the normal process. Now let's look at this a different way with a different picture. All right, here I've got some things I need you to memorize, and I've got a different way to try and explain this. So I need you to memorize with an action potential that it is self-propagated and it's all or nothing. You should also probably just memorize that it's associated with voltage gates. Okay, now the resting membrane potentials have a charge. In the sarcolemma, just memorize that it's -90. In your neuron, Most of the time, not always, but most of the time it's negative 70. But there is a wide range of variation depending on where you are in the body. So it can go from negative 20 to negative 100. The safe bet is just to remember negative 90 with the sarcolemma and to memorize negative 70 with your neuron. If you do that, you're safe. Okay? Now, let's look at this particular graph. And I want to point out that on the x-axis, you have time in milliseconds. So 0, 1, 2, 3, 4. Over here on the y-axis, you have your membrane potential in millivolts. Now, this is important right here. I've got negative 65. That is my, let me actually zoom this up. That is the threshold. All right, in order for a voltage gate to open, you have to change the voltage to hit negative 65 millivolts. Negative 90, we're going to breathe that RMP, resting membrane potential. This is where you are most of the time. So you're at resting membrane potential right here. Then what's going to happen is right here, you're going to open acetylcholine gates, so chemical gates, and that's going to let sodium rush in. All right, the sodium rushing in begins to change the polarity as it depolarizes. Okay? Now, if it only changes the polarity to there, everything stops. If it can't get past that point, if it only gets to negative 80, you do not get an action potential. It fizzles out. But if you get to right here at negative 65, you cross threshold. Now, right there, that opens voltage gates. When those voltage gates open, tons of sodium rush in, and that causes this huge spike. It's going to go way up. Okay, let me erase all those because that looks stupid. All right, so this is the major wave for depolarization that is creating your action potential. Okay, now we'll talk more about this point later on and why this happens, but essentially you peak at the voltage change. And this has to do with your sodium gates are now going to close and the potassium gates are going to open. We'll talk more about that later. As you do that, that begins to bring the polarity back down. So this is called repolarization. It's going to keep on going. It's now going to pass threshold. It's going to come down here. Now I want you to notice right here, all right, it's actually lower than resting membrane potential. This is called, let me change the color to something that's easier to see, the undershoot. All right, and this is basically because too much potassium leaves the cell. And then it's going to recover and it's going to go back to negative 65. And then you're at your regular resting membrane potential again. Okay, so that's the basic step. Now let's talk about refractory periods. All right, so I'm guessing most of y'all can get an idea of what a refractory period is, but in case you need it defined, a refractory period is when it doesn't matter what you do, you can't stimulate it to do it again, okay? So basically it's a period of time where you cannot generate an action potential, you cannot generate a muscle contraction. Now with muscle contractions, we have what's called absolute refractory period versus relative refractory period. Absolute, nothing you will do will ever result in a stimulus. It can't work. We'll talk more about this later and with neurophysiology, and we'll talk about how the gates just flat out can't be opened until they reset. Relative refractory period has to do with the membrane potential being hyperpolarized due to that undershoot. And you technically can overcome it, but you got to have a higher than normal stimulus, okay? So if we look right here at our little picture, we get our initial stimulus here. We'll switch that back to red. All right, we're going to hit the threshold right there, and we're going to generate an action potential. We're going to come up here, and then we're going to come back down here. We're repolarizing, okay? So right here, they're showing the absolute refractory period. So they've got it labeled up here. And basically, you've created a cascade of events, and you can't get those events to start over until they reset. And this has to do with how the gates are going to open and close. The relative refractory period, which is right here, this is due to the undershoot. And basically, your undershoot is a hyperpolarization, where you make it even more negative than usual. And again, you can overcome this. You just need an even bigger stimulus in order to do that. And then you get back to resting membrane potential, and you're back all to normal, and you can start and do the next thing however you want to. Okay? So those are your refractory periods. Now, the next section, we're going to get into part 6, excitation coupling. And we're going to really start pulling all of the pieces back together again and trying to build a full picture with the sliding filament mechanism, the neuromuscular junction, and excitation coupling. All right. And again, draw it out. Draw it out many times. It will make a difference. All right. So I'm going to stop there, and you can take a break.